Good afternoon. Welcome to our AHDB Soil Improvement Webinar 2. This is the second in a series of 3 soil improvement webinars. This webinar is on soil disinfestation. My name is Grace Choto. I'm an AHDB Knowledge Exchange Manager for vegetables. A few housekeeping points. You're all muted so that we don't pick any background noise. If you've got any questions, please don't hold on to them. You can type them in as we go. I'll show you how in the next slide. This is being timed and we will be through by 5 p.m. today. We are recording it so we can make it available on our website. For basis and Nerosso points, you will need to contact Maya, details on the next slide. And if you are tweeting, please, it's at AHDB Hort. Again, for questions, what you'll see is that on your screen, you've got a band which you can either maximize or minimize using this red arrow. If you scroll down, if you go down on that box, there's a questions box where you can type in your questions as we go. Questions will be answered at the end of each presentation. If you need basis and Nerosso, please contact Maya at maya.kotecha@ahdb.org.uk. The recording will be made available on our events archive on the AHDB Horticulture website. So this is about soils. Just to reiterate, we might have some people who joined us for the first webinar. AHDB has its own program of work called Great Soils, but also we're facilitators or disseminators of information arising from an EU Best for Soils project, which I'll touch on and which will be touched on again in this webinar. So Great Soils, if you Google Great Soils, it will bring you to our website, and on there we've got soil publications, videos. We've also got research project reports and a whole load more. You only need to type in what you're searching on soils and it will pull something out for you. At present, we've got 2 big pieces of work. One of them is on soil biology and soil health. It's a 5-year interlinked, it's got many projects within it. It's a 5-year program, and essentially it's because we know a lot about soil chemistry and soil physics. It's trying to really address those gaps that we don't know about soil biology, and then so we can link all together, you know, and to work to improve our soil's health. We've got also another 5-year program, which is called the Rotations Research Partnership. And in this project, we're looking at rotations with potatoes in them, and they're trying to make rotations more agronomically and really more efficient. So if you need to find out more, if you're interested in this work, please go on our website and you can Google these partnership programs of work to get more details. And that's just a sample of publications that you will be able to find on our website. So moving on to the EU Best for Soil project. This is the EU Horizon 2020 network project with 8 project partners who are applied researchers and advisors across Europe. In addition, there are 20 facilitators. We as AHDB have a facilitation role which ends on 30 September 2021. The project aims to improve soil health across Europe through promotion of 4 best practices and sound crop rotations. And our role as facilitators is to promote what, you know, outcomes and publications that are coming from this project. It's got free databases, fact sheets and videos in 22 official EU languages. And that's the main project webpage. So you just go bestforsoils.eu or you just Google Best for Soils and you should see it. And as you can see, it says that we're a network of practitioners trying to share the best knowledge available on the prevention and reduction of soil-borne diseases. And through the adaptation of optimized crop rotations, the use of cover crops, compost, green manures, biosolarization, and anaerobic soil disinfestation, which we'll touch on in this webinar. And those are the 4 best practices, including, that's where the 4 comes from in Best for Soil. We've got the pathogens and a nematodes database to help with crop rotational planning. Again, if you go on the website, you'll see a lot of publications, fact sheets, videos, and the 2 databases. And we'll do sessions on the databases in future webinars. So keep on look out at the AHDB events page. On Fusarium, for the program today, we've got 3 speakers. Our first speaker is John Clarkson from Warwick Crop Centre at the University of Warwick. John will be speaking on Fusarium diseases, understanding pathogen dynamics, diagnostics, and control in highly infested soils. John will be followed by Matt Back from Harper Adams University. Matt will be speaking on biofumigation and anaerobic soil disinfestation. And last but not least, we'll have Erika Wedgwood from RSK ADAS Ltd. And Erika will be speaking on soil disinfestation methods for the control of soil-borne diseases. So just to introduce our speakers, our first speaker is John Clarkson. John is a plant pathologist at the University of Warwick. He works at the Warwick Crop Centre, which is in the School of Life Sciences. John leads a group carrying out basic, strategic, and applied research in work funded by the government, research councils, and industry. He specializes in pathogens of vegetable crops, with particular focus on fungal soil-borne diseases caused by sclerotinia and Fusarium. John also works on disease management approaches, such as biological control and disease forecasting, which are needed for integrated disease control programs. He has over 20 years of pathology experience working with industry and other researchers through collaborative projects. He has led a large number of AHDB projects, most recently on Fusarium diseases of lettuce, which we'll be speaking on, onion and narcissus, as well as on cavity spot disease of carrot. He is a member of the Scepter Plus program, which some of you will know about, but this is a large screening program where we're screening actives and bioprotectants, biologicals, for efficacy against pests, weeds, and diseases. John will be followed by Matthew Back, who is a reader in nematology, Harper Adams University. Matt teaches plant pathology and crop protection to undergrads and postgrad students at Harper Adams University. His research focus is on the biology and management of plant parasitic nematodes, particularly on potatoes. His work on alternative management strategies for nematodes has been on biofumigation, and the outcomes from his research are being used to inform best practice in UK potato production. Matt also supervises PhD students, having current and previous involvement in 19 projects, and he's a co-investigator in the SARIC project, assisting with the development of an AHDB PCN model. Our final speaker will be Erika. Erika is a plant pathologist at RSK ADAS Ltd. She's an experienced pathologist since the 80s across a broad range of crops. Her current work is on soft fruit and ornamentals, but she's worked on recent AHDB projects, which include the evaluation of non-chemical control measures for verticillium and Phytophthora in strawberries and raspberries, and in investigating the efficacy of bioprotectants on protected crops. She's also worked within the soil biology, soil health project on the use of organic amendments on onion and narcissus crops susceptible to Fusarium. Erika's interest is in integrating the understanding of plant pathogen and pest biology into crop protection strategies. So let me hand you over to our first speaker, John Clarkson, who's speaking on Fusarium. Over to you, John.
Okay, good afternoon, everybody. Can you hear me and can you see my slides?
Yes, we can.
Okay, thank you very much, Grace. Thank you for the introduction. So that's the title of my talk. So I'm going to be talking about Fusarium wilt in lettuce. And I guess my job here as first speaker is to set the scene for a pathogen that is particularly challenging. And and one which we might want to control with soil disinfestation approaches. So just as an introduction to Fusarium oxysporum, then it's a globally distributed soil-borne fungal pathogen, and it causes vascular wilts and root rots of a wide range of different plants. So the main thing to remember about Fusarium oxysporum is that it's a species complex. So if you like, there are many subspecies or special forms, as they're called, forma specialis, and each of these is uniquely adapted to infect a certain crop host. So there's no cross-infection between these different forma specialis. And why is Fusarium a particularly challenging pathogen? Well, it produces these long-lived spores. They're called chlamydia spores, and these can survive for many years in the soil. The other challenge is that through deployment of resistant cultivars, Fusarium is quite good at evolving, and so it produces races that can break this plant resistance. And this can happen a number of different times as different resistances are deployed, as we'll see later. The other sort of challenge to do with the Fusarium oxysporum complex is, as well as all these pathogenic forms that you have, there are also non-pathogenic Fusarium oxysporum which are abundant in soils. And in fact, some of these are beneficial, and one has even been marketed as a biological control agent. So not all Fusarium oxysporums are bad, but it does mean that there's a challenge here for correct identification. Okay, so I'm going to focus on this talk on Fusarium oxysporum forma specialis lactucae. I'm going to call that FOL. It's present in most lettuce growing areas of the world, and there's a number of different races, and these have emerged, as I said, due to the deployment of resistant cultivars. So race 1 is the most widespread, and it causes disease in field-grown lettuce, mainly in warmer locations of the world, such as the USA and Southern Europe. There's another couple of races which are only found in Asia, so those are less important. But what's relevant to us in the UK is this very recent emergence of a new race, race 4, that has emerged in Northern Europe. And at the moment, it's only affecting protected lettuce, but has been particularly devastating, particularly in Holland, where a number of growers went out of business because of this new race. So how do we identify these races? Well, the traditional way is by doing plant tests on differential cultivars, as they're known. That allows you to identify races which basically infect different cultivars. So just a bit of information on the emergence of race 4 in the UK. It was first identified in 2013, so there's a paper a bit later than that, so it's kind of identified retrospectively. We first saw it emerge firstly in Ireland in 2016, and we confirmed in 2017 here at Warwick that we had this race 4 in both the Republic of Ireland and also initially in Lancashire. And since then, as you can see, it spread to a number of different sites, further sites, both in Ireland and in the UK, mainly obviously in those protected lettuce growing areas of Lancashire and Cambridgeshire. Not surprisingly, the varieties affected are the little gem and butterhead types, which are extremely susceptible. So the race is on with the breeders to produce a resistant variety to race 4, to FOL4. Just to reiterate, at the moment at least, no race 4 or any Fusarium, in fact, has been found on outdoor lettuce in the UK. But of course, there is a potential there for problems to occur later on in the field. So we had a small AHD project just to have a look at some aspects of FOL4. And this is what we were looking at. Firstly, how do we correctly identify it? It's quite important to identify the race because that will affect cultivar deployment. There were some questions about the conditions for disease development, which I'll cover. And also there was the question, well, if you leave your production area fallow or you put in another crop, will that actually reduce pathogen inoculum? And then through Septa Plus, we did a little bit of work on control measures. So I'm going to go through those now. So firstly, on identification of FOL4. Well, I think most growers now can recognise the symptoms quite easily. They're generally quite clear. You can see in the photograph here, very wilted lettuce, and they quickly die, and the leaves die back. The most characteristic, if you like, diagnostic symptom, which you can see in the photograph on the far right, is if you dissect your lettuce longitudinally, you can see this typical vascular browning. And it's generally, if you see that, it's very likely to be Fusarium. So we can confirm this in the lab. We can isolate from those plants, and in fact, we've developed a molecular PCR test specifically for race 4, which was developed through the AHDB project. And this test is also quantifiable. So we're beginning to use this test to see whether we can estimate the amount of pathogen in soils, and we've used it for other research purposes as well. We've also developed a very rapid test for FOL4 based on similar technology to PCR. It's called LAMP. This slide serves to illustrate that. So LAMP is, as I say, similar to PCR. You can use a portable machine though, and you get the results a lot more quickly. And in fact, what you can do if you have some infected lettuce, you can dig out a piece of tissue, shake it up with some reagents in a tube with a ball bearing, add some primers, insert it into the machine, and then you can get an answer in about 20 minutes. So in principle, those sort of tests can be deployed actually on farm. Okay, so that's how we aim to address the diagnostics. So one question that growers had was the effect of temperature, because they were seeing that Fusarium disease seemed to progress even in the winter months. So this is a question that we tried to answer. So simply growing isolates of race 1 and 4 on agar showed no difference, but actually when we infested compost with race 1 and race 4, and we put those plants at different temperatures, we found that actually race 4 could cause much more rapid wilt disease development and vascular browning, even at temperatures as low as 12 degrees C, as you can see in this graph here. So the graph just shows you the amount of vascular browning, the amount of disease, and you can see it's far higher at these temperatures between 12 and 20 degrees C for race 4 compared to race 1. And race 1 really doesn't get going until the temperatures get quite high, around 24 degrees. So we believe then that this race 4 can operate at much lower temperatures. The other thing we're trying to do is to establish what levels of inoculum you need in the soil to actually cause significant amounts of disease. So this is an experiment, actually it's still currently underway, so this is just initial data, but we've got a number of different soils here that we got from lettuce growers, as well as a peat-based compost that we use routinely, and we've inoculated that with different amounts of Fusarium, as you can see in the legends here, up to 2 million spores per gram, and not surprisingly you get a nice dose response, such that obviously high levels of inoculum lead to high levels of disease and more rapid disease development. But we can begin to sort of establish some thresholds for disease. So anything around 2,000 spores or below, disease progression's pretty slow. And what we're trying to do now is relate these results to the results of our qPCR test. So if we do a qPCR test on soils, we will be able to hopefully estimate what the risk of disease occurring is. So as I said, one of the other questions growers have is, what happens if I leave my soil fallow for a while? Will this effectively reduce inoculum of FOL4? So we did some tests. So we inoculated a polytunnel soil here at Wellesbourne. And we grew some lettuce, and then we took what we're calling a pre-fallow sample here. This was back in 2018. This is a highly infected crop, and we ran our qPCR test, and you can see this has high amounts of pathogen DNA. We had a winter crop following this, which actually didn't develop too much disease. It was pretty cold conditions. And then from the following January, we left this particular area fallow. And you can see from this top graph, there's a big drop in the amount of pathogen between this pre-fallow sample and when we started the fallow period in January 2019. So this bottom graph is really a blown up picture of this line from the top graph. But you can see actually, although the amount of inoculum declines quite rapidly at first, we can detect it for up to a year later in the soil. So although there's a decline, the pathogen is still maintained. And we also did agar tests. You can see the graph on the right-hand side here, which showed a similar result. More recently, we've actually grown lettuce in that tunnel, which was left fallow for 1 year and 7 months in total, and you can see that disease developed. So leaving these areas fallow doesn't seem to be a particularly practical solution. We also looked at non-host colonisation by race 4. So here we grew plants in infested compost to look at, and then we used our qPCR test to investigate how they were colonised by FOL4. Again, kind of bad news really on the control front, because we found that the roots of many different crop plants, which you can see along the bottom of this graph axis here, were actually colonised by the pathogen. So it's not causing disease on these plants, but it can colonise the roots. More interestingly, if we look on the left-hand side of this graph, we have a couple of lettuce lines here at Warwick, which are resistant to race 4, and yet the roots are colonized colonised, probably to about the same extent as other plant hosts, maybe a little bit higher in some cases. And if you compare this, and I'm just highlighting this, hopefully you can see this, with some susceptible varieties, you can see susceptible varieties obviously a much greater degree of colonisation. So you've got to be rather careful about deploying resistant varieties, although, you know, when these come, they may not express disease. It doesn't mean that the pathogen is going to go away. And this photograph at the bottom here actually shows you a partially resistant lettuce. So this is one which is commercially available. And you can see there is some vascular browning. So although there was some sort of marketable crop out of this, it's still being colonised. So finally, the other sort of control approaches we were looking at, which is related to disinfestation, was firstly looking at disinfectants. Hygiene is very important for control of spread of FOL4, you know, particularly talking about equipment and tools and propagation trays and things like that. So this table just shows you the survival of spores of FOL4 with different disinfectants that you can see in these different columns. And we had different durations of exposure, as you can see on the left-hand side, and different rates of the manufacturer's recommended rates. And all of these disinfectants that we tested, as you can see there, were fairly effective. One thing to note, though, is that some of these disinfectants may lose activity if there's a lot of soil contamination. That's something we were unable to test in the time that we had the project. But I can tell you that those based on quaternary ammonium compounds, according to the literature, do far better if there is soil contamination compared to some of the others. So the other approach, again, for disinfestation is to use heat. And we established in our project that actually 60 degrees is effective in eliminating FOL4 spores, providing you use that for more than about 5 minutes. So this is a graph. You can see, again, different exposure times along the bottom and different temperatures. So actually, hot water treatment, for instance, of trays or equipment for a fairly short period would be effective then in killing the spores. And we also tested disinfectants and heat treatments on polypropylene. These are polypropylene discs. So they're, you know, it's effective. These treatments are effective on sort of polypropylene type surfaces. So finally, on the control front, just to outline some of the work we did in Scepter Plus. This was a couple of years ago now, and we tested a number of different biological and conventional fungicide products for control of Fusarium, and this is what this graph shows. So again, what we're seeing here is the amount of disease measured as vascular browning score at the end of the experiment. And unfortunately, there's only a few treatments which we found to be reasonably effective. So firstly, Bazimid, which is, of course, a known soil sterilant, and there's issues there, of course, with its continuation. These other products in green did show some reduction compared to the inoculated control treatment, which is shown in red. Obviously, some of these are quite small reductions. But one of the biocontrol agents, T34 biocontrol, we found to be particularly effective. And you can see that in the photograph in the right-hand side at the top. You can see treatments with T34 and Bazimid indistinguishable from the uninoculated control. So lastly, I'd like to say something about resistance to Fusarium in lettuce. Obviously, that's still a good approach for control, with the proviso that you may still have the pathogen proliferating, or at least surviving in the soil. So we've got a big collection of lettuce at Warwick, and we screened these against race 1 and race 4, and we've identified some high-level resistance to race 4. And this is being developed in collaboration with Endsars, some of which is through an AHDB-funded PhD student, which is Andrew Legg, who you can see looking lovingly at some lettuce on the right-hand side. So these are the kind of results we can get. So these are a large number of lettuce lines, about 50-odd lettuce lines along the bottom. We're using lettuce of different types, not necessarily suitable for indoor cultivation, but resistance identified in another lettuce type can be introgressed into the butterheads. But you can instantly see the problem that the breeders face. Butterhead lettuces are essentially those that are grown in protection, and there's very little resistance in those particular lines that we tested. There's a few here that may show some promise. And just to show you some photographs on that, it does seem to be complete resistance. So these are lines which are resistant to FOL1 or FOL4. So we have uninoculated lettuce at the top and inoculated lettuce at the bottom, and you can see they look pretty much the same. And here is the line which is resistant to race 4 but not race 1, and obviously you can see the race 1 inoculated lettuce here showing very good symptoms of disease. So just in summary then, diagnostics can be particularly important to identify FOL races, and we're hoping that they may also help predict disease risk. We've shown that FOL4 causes disease in lettuce at lower temperatures than FOL1. There is a problem of these chlamydospores surviving in fallow soil for several years, but they do decline over time, and we're trying to establish the levels which cause disease and relate this to our molecular diagnostics. Non-host crops and resistant lettuce varieties may allow persistence of race 4. Heat and disinfectants seem to be effective hygiene methods, and Bazimid and some biological control agents may offer other approaches to manage FOL4. And finally, there's still an ongoing need then to develop new FOL-resistant lettuce lines. So just to thank a few people who've been involved in the work: Nicole, Sasha, Alison, and Andy. And thank you for AHDB for the funding and for your attention this afternoon. And I'm happy to answer some questions.
Thanks, John. I'm looking at the question box here, but I can't see anything. So if you've got any questions, please type them in. John, some of our listeners might not be aware that we also fund a bit of testing, so where growers have suspect samples. So were there any cases this year of lettuce Fusarium, and should outdoor growers be worried at all? Because it seems at present it's a problem only under protection.
Yes, we did have a few samples in from sites that I believed haven't had Fusarium before, so it is still spreading. In relation to outdoor lettuce, that's a difficult question. I think obviously race 1 is a problem in outdoor lettuce in other parts of the world and warmer parts of the world, but we have shown, or we have an indication at least, that race 4 can operate at lower temperatures. So in some respects, there's no reason why it shouldn't, you know, potentially be a problem in field grow lettuce. Having said that, there's, you know, protected lettuce production is a very different kind of production system. It's very intensive, and a lot of growers will just, you know, have lettuce crop after lettuce crop after lettuce crop, and it could be that the breaks in the field are enough to, you know, to prevent race 4 from being a problem in the field. But, you know, I think it's early days. The pathogens only really just arrived in the UK, and we've seen situations previously. For instance, unfortunately, I'm old enough to remember what the major problems in onions used to be: white rot disease, you know, 15 years ago, and now it's Fusarium. So, you know, Fusarium in outdoor onions has become an increasing problem, and that could be related to climate change and things like that. So we could see that situation in lettuce with Fusarium.
Thanks, John. We've got a question here. Is there a relation between lettuce Fusarium and spinach Fusarium?
The answer to that is no. So the spinach Fusarium, as it were, is a different form of specialis. It won't affect lettuce, and vice versa. But as I showed, you know, some other crops, and I think, you know, spinach was on the list there. Spinach can be colonized by race 4, but it doesn't cause disease. So there's a difference. There's a difference there.
Thank you. We've got something, you've already answered this. Why does this pathogen not affect outdoor plants? Is it because of the cold weather? And I think you have partly answered it with rotations.
I'm not sure temperature's a barrier. I think it's more a case of maybe it's just not got out there yet, or the rotations are stopping it becoming a problem.
Well, thank you, John. John actually has a sore throat, so we were worried whether he would be able to, and a cough, make this presentation till the end. Thank you very much. If there are any further questions and you're available, John, then we can answer those at the end. Thank you. We are moving on now to our next speaker, who is Matthew Back from Harper Adams University. Over to you, Matt.
Hello, can you hear me, and can you see my screen okay?
Yes, we can.
Perfect, okay. So good afternoon, everyone. My name's Matthew Back, and I work at Harper Adams University. I was asked by Grace to talk to you about some of our work on biofumigation, and requested that I include a small amount of information on anaerobic soil disinfestation. And I have to confess that we haven't done any specific work on this, but I'm going to be running through some basic information on what anaerobic soil disinfestation involves. Okay, so just to give you a brief outline of what I'd like to talk to you about today, I'm going to be just going through biofumigation in general. Some of you who are listening to me may have heard me talk before, or indeed heard others talk on this topic. I just want to introduce you to that topic, and then I'm going to be giving you a little bit of our own evidence of activity. Now, a lot of this work has taken place in potatoes, but can be applicable to other crops and other problems. And then I'm going to be talking about the practical use of biofumigation. So what I want to do here is to sort of give you something to take away in terms of how you might use this technique practically. and then I will finish with a very short overview of anaerobic soil disinfestation. Okay. So first of all, just a few questions about biofumigation. First question is, what is it? And effectively, biofumigation it's quite a broad term, but it refers to the use of naturally occurring volatile compounds for the suppression of pests, weeds, and pathogens. And most of us that are probably listening to this right now will associate biofumigation with the use of brassica cover crops, but you can get biofumigation from other types of plants and indeed fungi as well. The next question is, why are we interested in this process of biofumigation? And the answer is that over the years we've seen a lot of loss of products, and if we go back in time, there was the phase out of methyl bromide under the Montreal Protocol of 2005. Following that, there's been a lot of losses of synthetic fumigants, and researchers and the industries in general have been looking for an alternative to replace those synthetic fumigants. And in addition to that, we've had the big movement on soil health and cover cropping in the last 10 years. And biofumigants are seen as part of this profile of cover crops that could be used, well, mainly for pest and disease suppression, but there are also added benefits of having these deep-rooted brassica crops for drainage and also for increasing organic matter and so on. How about the UK? So worldwide, there are a number of different applications of biofumigation, and there are commercial products that you can use. You can buy mustard meals, for example, granules that you apply to soil, typically sold as a fertilizer, but they work as biofumigants. There are liquid formulations as well, but in the UK, our typical approach is to grow what we call glucosinolate-rich brassica cover crops. So these consist of crops like Indian mustard, oilseed radish typically, and these are grown till they reach early flowering where the chemistry is at its maximum, and then the biomass is chopped and bruised as much as possible before being buried, and ideally that material should be buried into moist soil, excuse me. So this is a lovely picture of a biofumigant being grown locally in Shropshire. It's Indian mustard, and on closer inspection in this field you could find all sorts of pollinators and insects. So there's another good reason for having a biofumigant. So the basic premise of biofumigation is the chemistry that you find in these plants, and everyone has experienced biofumigation, whether you've grown biofumigants or not, and that is that we've all tried mustard. A lot of us have tried horseradish, and we know the hot, pungent flavors that come from these products, and that's down to glucosinolates being converted into volatile compounds. So if you take too much mustard, you know that shooting sensation that you get, and that's all to do with what's going on in biofumigation, the chemical process behind that. So we're trying to grow these plants for glucosinolate content. This is the important metabolite. The glucosinolates themselves are not the bioactive compounds, but they are the precursor to the bioactive compounds. So we grow these plants to maximize glucosinolate content, and typically this occurs around the point of early flowering. This is where they peak, and if we crush these plants through chopping or bruising, then we'll cause the release of the glucosinolates, and at the same time, in separate cells in the plant, will cause the release of the enzyme myrosinase. And in the presence of water, so water coming from the water from the plants within the vacuoles or from the soil moisture, the glucosinolates become hydrolyzed to produce a range of different volatile organic compounds. And there's a whole series of these, but the compound that we're mostly interested in is the isothiocyanates. These are the largest molecules that are produced, and they're more persistent than the other volatile organic compounds. The type of volatile organic compounds is dictated by the pH of the soil. And ideally, with biofumigation, we want a soil which has a higher pH, ideally between 6 and 7. If it starts to become low, so if we have an acidic soil, so if we have 5 or below, then we tend to get the production of other volatile compounds, namely nitriles. And whilst these are still biocidal, i.e. they would have an effect on your pests and pathogens, they don't persist as long, so their effect is not as great as isothiocyanates. I should also mention at this point that it's not ideal to have a soil that has high organic matter, because a lot of these volatile organic compounds can be absorbed into that organic matter. Okay. Now, there are 3 stages to biofumigation. The first stage is what we've termed as partial biofumigation, and that's the effect of the growing crop. And this is an interesting phenomenon that happens from these plants, in that they will naturally leach glucosinolates as they grow, and we've detected glucosinolates beneath the roots. The question is, how do those glucosinolates then get converted into isothiocyanates? Well, with a bit of research, we found that there are a range of different microorganisms that are present in the soil that will actively use the glucosinolates, because they are glucose-based molecules. And in converting or trying to access the glucose by breaking down the glucosinolate and using myrosinase, they inadvertently produce the volatile compounds. So even before you chop the crop, you can get a biofumigation effect. And this is interesting. This is something that we've wanted to do some more research on for a while, because we feel that the next stage, which involves chopping and incorporation, whilst it's very effective, it's often done during the winter months, and that can be damaging to the soil itself. So a system whereby you could look at the growing crop rather than the chopped material is interesting. So full biofumigation, as I said, involves chopping and maceration of tissue to release all those compounds and expose them to water to get the production of the volatiles. And then the third stage is what's known as post-biofumigation, sometimes referred to as the organic effect. And this is simply that we're adding a lot of organic matter to the soil and a lot of carbon sources, and these carbon sources may be used by microbes that are antagonistic to pathogens, and this has been shown in some work that was conducted in Australia. Okay, so just to show you a little bit of evidence of pest suppression, this is on a pest that I've worked on fairly extensively, and that's potato cyst nematodes. So PCN nematodes survive in the soil as the old females, the cysts, and these contain up to 600 eggs. In this particular study, we've got 4 treatments. You can see here, this is the measurement is the number of viable eggs per gram of soil. The treatments are 3 different biofumigants that you can see down here, and that's rocket, radish and Indian mustard, and this is compared to an untreated fallow soil. So this initial one is just the initial population before the biofumigants were sown, so there's no significant difference there. But pre-incorporation, just before those the crops of different biofumigants have been macerated and incorporated, there is a suppressive effect. And similarly, we can see the effect, and with Indian mustard to a greater extent, after the material has been chopped and incorporated. And if you know anything about PCN, you'll know that if you grow potatoes, you'll multiply the nematodes, so you end up with a higher population after potatoes. And what we can see here is this was surprising that we get a legacy effect of biofumigants, and the significant effect is with Indian mustard. So here's just a little bit of other evidence that we've got on biofumigants working. We can see here this is Fusarium graminearum. This is a pathogen that causes Fusarium ear blight in wheat, and it's important because of fungal poisons, mycotoxins. And this is just looking at the active ingredient of biofumigants. This is actually the active ingredient of Indian mustard, which is allyl isothiocyanate, and we can see this suppressive effect with raising concentrations on the colony growth here, where we've got the highest concentrations up here. We've got no growth of that fungus. Similarly, sorry, if I just go on to the next one, similarly we can see with high dose of this isothiocyanate here, we are getting inhibition of the germination of the spores, the conidia in this case. And this is a graph which shows, it's a complicated graph, but it shows how the radial growth of the fungus is affected in different ways by different isothiocyanates. So the use of this type of study is important in the selection of the right type of brassica for biofumigation, because we know what are dominant glucosinolates, which then go on to give the relevant isothiocyanates. We can select those in this type of process. And this is a slide, this isn't very empirical at all, but it's nice in that it shows you a similar radial growth experiment. So we've got Fusarium graminearum, and all we've got in these plates is different numbers of discs, leaf discs of Indian mustard. And these leaf discs have been flash frozen and then allowed to thaw, which then in the thawing process, this releases all the volatile compounds. So getting the most out of biofumigation. First thing to think about with biofumigation is two things. What we're trying to do, we want quantity, so we want as much material as possible in terms of biomass. That's the amplifier of the process. Secondly, we want glucosinolate content, but we want the right type of glucosinolate content. So this comes from knowing what species produce which glucosinolates, which then produce isothiocyanates that are most active. There are some glucosinolates that are found in plants, such as indolil glucosinolates, that have no activity, that don't produce isothiocyanates. So it is important to select variety and species very carefully. So in doing so, thinking about species and cultivar, I've mentioned Indian mustard, which we can see here. This is rocket, and this is oilseed radish, and this is white mustard. In all of our studies, I have to say the most consistent performer, and when you look at the literature, you will find that Indian mustard is most consistent in performance. However, we have had some good results with oilseed radish as well. White mustard doesn't produce the right profile of glucosinolates to have activity in biofumigation. So really we shouldn't be looking at white mustard. In terms of crop management, first of all, think about the site selection. Remember what I've said about pH. Soils with very low pH possibly going to be less effective. Also, soils which have got high organic matter content, you could get more absorption of the isophyocyanate, so less activity there. A question that comes up is, when should I grow my biofumigants? And in all the work that we've done, the answer is, if you can, make the most of the summer months. So going after carrots, for example, going after winter barley, these are ideal crops to go after. So they're sort of looking at sort of a July sowing date, and then incorporating at the end of September, October. So typically a crop of Indian mustard would grow for 12 to 14 weeks. So using those summer months is important. And when you look at the literature, it makes sense, because glucosinolate concentration is affected by UV in terms of the day length, the intensity, and so on. So it is important to use those longer days with high UV intensity. Nutrients, two key nutrients that are important are nitrogen and sulfur. We'll come on to that in a moment. And the cutting time, I would, as I suggest, earlier the better. So going into October rather than November, where temperatures are starting to drop. If you've got a soil temperature below 8 degrees, this is not suitable for the activity of volatile compounds. And I'll talk a little bit more in terms of implement selection in just a moment. If the soil is very dry, you could consider irrigation. And the reason for this is if there's a lack of water available for hydrolyzation of the glucosinolates, this isn't going to maximize the biofumigation process, and we would recommend a soil moisture content of 50 to 75% of fill capacity. This is just a few graphs which nicely highlight what nutrients do in terms of biofumigation. Here we can see the impact of the nutrients in terms of increasing sulfur content on glucosinolate concentration. Clearly going up to 25-50 kilograms per hectare of sulfur is beneficial, and as you would expect with this, if we look at this graph here at the bottom, we can see the effect of more so nitrogen affecting biomass. So when we put those two graphs together and we look at what's happening with PCN here, we can see that, and this is one I want to draw attention to, where we're applying 100 kilograms per hectare of N, so we're getting the biomass from there, and where we're adding then 25 kilograms per hectare of sulfur, we are getting a better suppression of our PCN eggs. So that's the take-home message. Okay, in terms of maceration and incorporation, we've got a number of different decisions that we can make. What type of equipment can we use? In this illustration, you can see with the blue bars the effect of comparing a flail versus a roll conditioner. A roll conditioner tends to produce larger fragments but more bruising. A flail tends to do more chopping. And in general, we found that the flail was more effective in reducing egg viability of PCN. When we looked at the implements, it's a bit complicated. If you look down at the bottom, you can see the comparison between a spader, a plough, and a rotavator. Spade is a deeper cultivating implement, and we were able to pick out a difference there when we're spading and when we're flailing, compared to if we were using a rotavator and a roll conditioner. And the comparisons that you've got on this graph are partial biofumigation, illustrated by the green bar, and an untreated fallow by the red bar there. Okay, so these are just a few effects of that you might want to consider with biofumigation. The first is should put you off growing Indian mustard during the winter. This is frost damage on the left-hand side. Some of these biofumigants are particularly susceptible to clubroot. Indian mustard, rocket, very susceptible to clubroot. However, something like oilseed radish is less affected by clubroot. But neither does it reduce clubroot afterwards. And also crops like Indian white mustard, very susceptible to cabbage root fly, which you can see there on the right-hand side. So just for a bit more information, do have a look at this guide here. This is for PCM, but would be useful to consider for biofumigation in general. Also have a look at Best for Soil, some useful information on there, as Grace has mentioned. I'm going to move very quickly onto anaerobic soil disinfestation, as I realise that my time is disappearing rapidly. So first of all, what is ASD? Anaerobic soil disinfestation is essentially creating anaerobic soil conditions for a limited period of time. And what we're doing is we're creating those conditions to cause asphyxiation and therefore mortality of the pests and pathogens in the soil. Now, there's two places where ASD has been investigated. First of all, in the Netherlands. So the Netherlands obviously have dikes around the fields and they can flood their fields, and this is called inundation, and this can be used to effectively kill pests and pathogens in the soil. The method that was developed in Japan involves the incorporation of fresh green material, could be grass, cover crop material or crop residues, and ideally 1 ton of fresh weight per hectare per centimetre depth of soil. So if you wanted to go to a depth of 40 centimetres, you'd need 40 tonnes of fresh weight per hectare. And here the soil should be wetted, ideally to fill capacity, pressed and then tarped with a virtually impermeable film or polythene, and that should be for 6 to 8 weeks, and the soil temperature should be 16 degrees C. And the idea is that we're trying to encourage anaerobic microbes that you can see in this slide that effectively, so different types of bacteria that suppress our pathogens. And these are all the sort of different things that we can do. Nematode problems, various fungal pathogens. And if you look at the Best for Soil website, you'll see loads of different things that you can do. The good and the bad with ASD, first of all, very, very wide spectrum of control. Lots of things can be reduced with this method, and there is some recovery of non-target organisms, which is good. And because you're adding this, all this carbon sources, you can increase nutrients as well. The big drawback for me is the cost. You'd only really do this for high value crops. Calculating the cost is £3,600 per hectare, which is a lot of money for the film, and you've got the labour. The other thing is that you've just got a very limited window for application due to the required soil temperature. You have to do this in the summer. There is some evidence of phytotoxicity, and some pathogens and nematodes are less sensitive, i.e. stubby root nematodes, Pythium, wart disease, which has thick-walled spores. And then the other downside is you will get the loss of some earthworms, springtails and antagonists. So very sort of quick wrap up on that ASD. If you want to find out more about it, do have a look at the Best for Soil website, where there's lots of information. And I'm going to finish there because I realised I've been talking for too long, and that's always a problem with me, is that I do talk. Okay, is there any questions?
Yes, they are. Thank you, Matt. Our first question is, the AHDB bulletin states that isothiocyanates do stimulate PCN eggs to hatch. Does this also happen with other cyst nematode species, e.g. melodygynous species?
it doesn't really stimulate egg hatch. What it does, Grace, is it has an effect on reducing the viability of the eggs within the cysts. And what we found is it reduces the percentage viability, but we also see a reduction in the number of eggs in the cysts as well. If it, as to whether it would have an effect on other cyst nematode species, then whilst I've not tested that specifically, one would expect that you would have similar effects. And with regard to root knot nematodes, again, not tested it specifically, but there is plenty of evidence in the published literature about root knot nematodes being suppressed by biofumigation.
The second question is, do you have any experience on the effect of biofumigation, mustards, and slug activity? So do biofumigants reduce slug activity or increase it?
I've not seen, we've conducted a lot of field experiments, and I have to say that I've not really noticed any, and we've not been specifically going out to look at slug activity, but I've not seen an increase in activity. When I first began reading all about biofumigation, one of the things that you read about very often is that these compounds are biocidal, which means they have an effect non-selectively on a whole range of organisms, including slugs. So one might expect that there could be some reduction of small juvenile slugs and eggs, but that I would just add the caveat that I don't have data to back that up, but that would be the expectation.
Thank you. How often in a rotation should you biofumigate?
How often in a rotation should you biofumigate? I think at least you should practice it at least once if you can, but it will not do any harm because every time you biofumigate, if I was to take PCN as an example, we've seen reductions from 45 up to 70% of the population using biofumigation. So I always think about biofumigation as taking out a proportion of the population. I don't see it wiping a population out completely. So if there is an opportunity to practice it more than once, then I don't see that as being a bad thing, but at least once.
Once in so many years? Once in a year?
A typical, you know, if someone had a 5-year rotation and they were looking at nematode control, particularly if they were looking at cyst nematodes, yeah.
And the last question before we move on is, can you do a test of the soil for PCN numbers when growing potatoes?
Can you do a test for the soil for, sorry, could you repeat that again?
Sorry, Matt. Can you test for PCN numbers, for potato cyst nematode numbers when you're growing potatoes? Is there a test for it? Yes.
Yeah. There's a standard soil test whereby you can extract the cysts, you count the eggs. We put a little bit more detail onto it when we're doing this biofumigation work, in that we run viability assessments as well, which are very often missed out, and we feel that they're important because you can count eggs but you don't know if they're viable or not. So we've got a range of different viability tests that we use.
Thank you, Matt. We're going to have to move on to our final speaker, Erika. But also, listeners, could you note that in the handout section, so on your screen, we've put the handout that Matt spoke about. He's written a biofumigation guide on potatoes, but it will be relevant to other crops as well. So have a look at in the handout section, you can download it. Over to you then, Erika.
Okay. I've just clicked show my screen and didn't get the PowerPoint up. Hang on, there we are. Oh, I've got to move this out of the way, sorry. All right, hello. So I'm going to be talking to you about soil disinfestation for the control of soil-borne diseases. So the outline of my talk is going to be starting off just covering what soil-borne resting stages can be found from different diseases, touching on integrated disease management. I'm going to mention chemical disinfection of soil, but the main essence of this talk is going to be the thermal disinfection of the soil, looking at some kit that is available as examples and the efficacy of the treatments, and finally touching on healthy soils and how we can approach those. So first off, survival as resting structures in soil and plant debris. The resting structures are thick-walled and they're formed as the host food source depletes. So they'll tolerate heat, freezing, drying and wet conditions. And particularly if they're inside slow decaying stems or roots, they can survive for some time. So in fact, this can be several years. So crop rotation can be impracticable. One thing to note though is if you're doing chemical or physical soil sterilizations, that 100% kill isn't guaranteed and is probably quite unusual. So some pictures there of resting stages developing. On this one, it's sclerotinia on stalks. Those little lumps are forming sclerotia. The Pythium oospores. This is chlamydospores of black root rot Thelaviopsis. And this is Rhizoctonia black scurf on an agar plate. So there's some various pathogens and the resting stages that they have. So if you have sclerotinia, I just said you have sclerotia. Verticillium also has microsclerotia. We heard that Fusarium has chlamydospores. There's also other pathogens that do. And then the oomycetes have oospores, Pythium phytophthora, but also the downy mildews, and also pathogens such as Septoria on celery will have Pycnidia. So I just wanted to look at integrated management just briefly to remind you that soil disinfestation is the last resort really. So you should be starting off with clean seed into clean soil. You may want to use a biological amendment. We heard about T34 from John. And then keeping an eye on your crops, and if there are any plants that are looking diseased, ideally to remove them and to work out what's wrong with them. When you come to harvesting, to remove the whole crop if at all possible. And if not, then if possible, remove the debris. And I'll be showing you a slide about that in a minute. And of course, general nursery hygiene. And if you do all these first steps, you may not actually need to get to the soil disinfestation need. So the first thing up here, we've got Basamid, Tazomet. I'm sure many growers have been using this product. From July, it was withdrawn from aspects of growing. AHDB have tried to get emergency authorizations again for use in soft fruit, ornamentals and salad leaves, but at the moment CRD has rejected that, saying that flame sterilization and steam sterilization can be used instead. They're still trying, so we'll see how that goes. So if we are able to use it, it's different from steam sterilization. It does need this moistness, and that's so that you can get the methyl isocyanate released from the product. It has got to be sealed for at least a week, and in contrast to steam sterilization, you do need to leave this period before you can plant, because of the fumes that are regenerated. So if we are using thermal treatment, what are the lethal temperatures and pathogens? One of the slides that John showed was the Fusarium lethal temperature. And as we can see on this diagram on the right, this was a test done by ADAS some while ago, looking at the different pathogens and the different lethal temperatures. So Fusarium is one of the more resistant, and these other ones are easier perhaps to deal with. So 65 degrees for 10 minutes can, in the lab in moist soil, just the spores on their own can cause death. But in the real world, in crop beds, you're going to get uneven heat spread through the soil, poor penetration of soil clods or the infested debris. and it will require heating at least to over 65 degrees for at least 30 minutes. And if you have got pathogens inside woody stems or in sclerotia, you will need probably over 90 degrees to be able to penetrate and to kill the pathogens. So I think it is important when you're doing this thermal work to actually have a thermometer or a probe in the soil, so you're aware of what temperatures you are achieving and for what duration. So this first picture is the flame weeding. Well, it is sort of marketed for flame weeding, but it can also be used for destroying small sort of soft crops like lettuce debris and that sort of thing, and that will stop the pathogens, if they're in the crop, making resting spores and dropping them back into the soil. So it's a hygiene measure. If you've got woody stems, there is a machine that will chop them up and flame them, and that again will reduce spores falling back into the soil. There is also a machine where you can have a rotavator to finish off your bed preparation, and then that flames the soil as it drops down again. But it will only do the top 5 to 10 centimetres, so it's not a measure if you've got a real problem in your soil. It's just if you want to get rid of the surface infestation. So moving on to other aspects of heat treatment, sheet steaming, of course, has been around for quite some time. You can see here it being laid out with the pipes coming from the steam boiler, and that green sheet will be pulled over and weighted down at the edge. So before you start putting that down, you will have made sure that the soil is fairly dry, so adjust your watering before you in the last crop. Wet patches can't be treated because the steam needs to push through the air. So any compacted areas, make sure they're spaded up and aim to cultivate for about 35 centimetres. Certainly if you've got a deep rooting crop, aiming for a coarse tilth, no wider than about 5 centimetre if you've got clumps left. You'll be using a heavy duty plastic sheet to cover the soil. Obviously make sure it hasn't got holes in, because you need to actually inflate it and make sure also that you've got your edges are weighted well down. It's a good idea to actually put these weights beyond the bed of the soil so that the pathways are included. Otherwise, I know I've seen infection coming in from the edges into the crop again. If you can find a thermal fleece or a foil, it's a good idea to put that over the sheet because it will keep the heat in. So there's two options. There's dry steam to 100 to 130 degrees C, 1 bar from a steam boiler, or you can use lower pressure steam generation. And with this one, you boost the steam to 140 degrees C with a superheater. So this is the sheet that's been laid out here. It will need quite a time to inflate, and it then takes 4 centimetres an hour to penetrate into the soil. So once it's actually inflated, it's about 6 hours to reach down to 24 centimetres. And you need to maintain a temperature, ideally over 90 centigrade, for 30 minutes. So that sheet will be kept raised for 4 to 6 hours, and then you'll leave it on until as the soil cools. So a full steaming cycle for sheet steaming is usually about 8 to 12 hours, and about 50 to 700 meters square can be sterilised at one go. Another method that was investigated and tested for the viability of Fusarium in the soil afterwards by ADAS was using a steam plough. And in that you can see there's the plough is winched along, the tines are digging into the soil, and along each tine is steam jets that then penetrate into 33 centimetres or so of the soil, and the steam goes along those channels that the tine is creating. So it goes along and up, and that does quite a good job at sterilising. So the actual heat is only on one particular point for about 23 minutes because the sheet, depending on its length, is moved off that position. And similarly, according to the width of it, this particular one, it only did 10 meters an hour to actually treat an area of 80 meters squared. So 7 hours, and that's quite slow, really. But what is the comparison between sheet steaming and steam ploughing? So the top graph here we had is the sheet steaming, and you can see that there's a fairly gradual rising in temperature. So at 23 or 24 centimetres, it's that 4 hours before you really get to the temperature that you want. And the lower one clearly doesn't make 100, so the deeper isn't sterilised as to a hotter temperature. Steam ploughing, we heard that the sheet is moving, so it does actually manage to inject it and get the hot temperature very, very quickly, and then it's taken off the sheet, so you will get that rapid drop down. Work done with burying bags at different depths of Fusarium of stocks did show that there was equivalent killing of the Fusarium in the stem, so between 90 to 98% of the kill for Fusarium in both these situations. They were very comparable. Another site was investigated for sheet steaming, and you can see that graph. It's not attained the temperatures that site 1 attained, and this top surface has dropped off very quickly. So that's clearly an issue. So you can buy thermistor probes. There's one here, digital probe, lactucae. There's probably other makes. So it's important you put these in so you know what your temperature is and that you've achieved what you were hoping for. Of course, if you haven't achieved it, you need to work out whether it was a soil moisture, whether it was too many big crumbs, or some other thing functioning with the steam boiler that was causing the problem. So here I've taken out those maximum temperatures from the steam boilers, not the steam plough, just the steam sheeting. And we've got the site 1, which is the good site, and the site 2, which is the poor site. So you can see that in the depth, these site 2 didn't make even 60. We just got about 60 at 10 to 15. So on the left here, on the right, sorry, is the percentage kill of Fusarium in buried stems in little bags. And the site 1, clearly you've got a good kill. As I said earlier, it's not going to be 100%. There'll always be something that somehow survives. But you can see right at the right here, the deeper ones for site 2, there was hardly any death. The best kill seemed to be at 10 to 15. Not so good in this one, possibly because that temperature fell off very quickly, and it's obviously harder to kill things in woody stems than it is in the roots. There's a better kill in the roots. So I said you need to be aware of what can happen if you don't heat it to the right temperature. And so this heating is important for any technique you're going to be using, not necessarily sheet steaming, but anything where you're putting the heat into the soil. You need to obtain those temperatures, 80 to 100. So one way of actually getting the heat further down into the soil is by using a vacuum or negative pressure sheet steaming. And in this you use a pump which draws the steam down, and there's a buried perforated pipe. If you have it temporarily, you can put it about ½ metre down. If you're actually going to permanently install it, you can have it 80 centimetres down, and that pulls the steam down from under the sheet into the soil. And so that measurements that ADAS took in this site here found that you could get 95 degrees centigrade after about 5 hours. But there was some measurements, say for example on the wall, that that temperature wasn't achieved. So maybe it was either the soil wasn't loosened enough, or perhaps it was by the wall conducting the heat away. So you do need to measure and make sure it's getting the heat at all positions. Hood or plate steaming, and this really, you can see these metal plates on both these models. It's really replacing the sheet, and the hood is forced down into loosened soil and left there for the steaming period dictated by the soil type and the depth required. The blue machine is a robot, so you just program it and it goes and moves the thing along. And I think the right-hand machine is a tractor towed, so it will be the driver working out how long to leave it at each particular position. And other models are done by this other company, Rigero, and you can, if you've got a small glass house or fairly small glass house, you can have an operated one where the man or woman stands on it, or you can have the robotic versions of up to 7 beds. And again, there has been a project on it, Field Veg 229, and that showed that soil type and moisture content can affect the penetration depth. And I think that's the same with any steaming, really. This last picture is of something coming it's quite vicious, doesn't it? Sandwich steaming, and these spikes are pressed down into the soil, and because they go down, they can actually do from the surface down to 25 centimeters, and that hood is left there for about 5 to 10 minutes, and then automatically lifted up with the hydraulics and then moved down onto the next piece of soil. So it is fairly quick, and you can do more area for the same time of sheet steaming. So this is a summary of the different methods I've just talked about. Sheet steaming, of course, is well established, and that will get down to 30 centimeters provided you've loosened the soil. Steam ploughing, that does allow the easier attainment of steaming to 30 centimeters because it injects the steam, but again, that is slow. Vacuum steaming, that's good because it does suck the steam down to at least 80 centimeters if you've got the pipes down there. Hood steaming, it really is just simplifying the sheet steaming. You don't have to be lugging those sheets around, and you can change the duration of the steaming to how much depth you want to penetrate to. And finally, that sandwich steaming, clearly that's a good combination because it gives both the depth and the surface steaming. So that's a good idea. There are other options that have been investigated. Microwaving is certainly been studied, hot air and direct heat treatment. There are models where you lift up the soil and then drop it down again, but it's perhaps more for glasshouse or for making compost. There are fact sheets from 2007 that are available, and that these also have details of the pathogen kill differences from AHD project 213. So just to say, after steaming, if you're not used to it, you can plant straight away after it, but it's best to wait a few days because the soil structure has been changed so much. And also to be careful when you start watering, it needs to be able to permeate again into the soil, not just wash and clog. Do be sensible about machinery and footwear. Don't bring stuff into the house again that you've already cleaned up or into the field. And similarly with cultivation, you make sure you don't cultivate up from below what you've sterilised. If you are steaming for the first time, there manganese can be released and cause toxicity, and often the heating will release organic nitrogen, leading to the release of nitrates. So a soil test is recommended for that, so that you know with your subsequent feeding what needs to be done. One thing I'd like to highlight that is, although weeds and plant infesting fungi, bacteria and nematodes are killed, so are beneficial members of the soil, microflora and macroflora. So some action points: do at the end of a crop either flame it off or remove the residue without scattering the spores everywhere. Make sure you know what depth of disinfection you need based on your past rooting and where these spores have been produced on the plant. So for example, if it's verticillium, they'll have been produced on the roots. And also your future crop roots, how much are they going to go down into the sterilised soil. Make sure it has a fairly dry, coarse tilth. Use thermistors to record temperatures. And remember, you should really be aiming for 90 degrees C with sheet steaming or any other steaming. And be aware that you have to learn about your own soil type because that will affect the heat transfer and how long you have to steam for. And then on the right, this is really hygiene, don't basically bring the disease back into the house. And if it is in there, keep an eye on it and work out what you've got and remove the plants if possible. So that's most people will know that anyway. So finally, I've got my final slide really is thinking about healthy soils. The reason that you've got infested soils in the first place is that one of the reasons that they've become out of balance. There could be conditions such as poor aeration, low organic matter or unbalanced soil chemistry, and that's affected the resistance of the plant to the disease and affected the microbiome. Because healthy plants, they have a rhizosphere of beneficial fungi, bacteria and macrofauna, and part of their healthy aspect is they have root exudates. And then the organisms feed on these root exudates. There's a whole food chain of organisms that feed on that original exudate, and these can actually produce like an army really against pathogens. Many plants also, if you haven't sterilised the soil, find other fungi and can develop a mycorrhizal system, and that will assist with water and nutrient uptake, and also plant defence compounds can be stimulated. You can actually buy microbes and various supplements to put in soil, and they have been shown in various experiments in certain situations to induce plant defence responses as well. So I think really soil disinfestation is a short-term mix, and growers ideally need to sort of nurture both plants and the soil and get a healthy soil going there so that you can get your plants growing strongly. So as we mentioned earlier, there is the Great Soils Project and the Soil Biology for Soil Health, and they've got information on achieving good soil health and information on the soil health card. So if you're outdoor cropping, to actually work out whether your soil is healthy or not. Okay, thank you.
Thanks, Erika. We've got a question here. What is your experience in controlling weed pressure with steam sterilization? And it goes on to say, given that the soil needs to stay drier in turn, which promotes seed survival. So I'll say that again. What is your experience?
I haven't got, the only experience I have actually is my personal one, which is a steam sterilisation of chrysanthemums in a glass house. So I'm afraid I can't answer that. So sorry.
All right, I'll move on to the next question. Has research been done on the use of trichoderma as a soil treatment after steaming to reduce reinvasion of soil pathogens?
Again, I'm afraid I can't answer that one. I don't know. Sorry.
The third one is, are there any beneficial microbes, biocontrols for the control of verticillium? I'm not sure. No, sorry, I'm not sounding very good for these answers, am I? The work I've done with verticillium has been using ASD, so Herbie products, and they did reduce the sclerotia in the soil. This was in the MOPS project. So that was a component, not actually microbes. And Erika, just a question from me as well. It seems quite an expensive method, but based on time and obviously heating, whatever you're heating to give off the steam, probably water. I don't know whether you put anything else inside it. In the studies that ADAS did in the research AHDB funded work, is the information on economics and cost of, if you do really need to, because your soil is out of balance, is there any costing information on per hectare? If you use this, it would cost you this much or not?
I don't think so. And it was 2007 as well, so it probably needs updating. I think it was mainly based on the pathogen kill rather than looking at the costings at that time.
Thank you very much, Erika. I'll just go to my next slide, please, Maya. Thank you. So just to say these things, well, thank you. Just to thank all our presenters today. Thank you, John. Thank you, Erika, and thank you, Matt. If you need any basis on ROSO points, you can contact Maya. Like I said, we've got handouts that are available in the handout section that you can print out or read online. If you've got any further questions, please send them through to me at ahdb.org.uk. The recording will be made available online next week. By the end of next week, it should be there. And again, look out for the last of this autumn's AHDB horticulture webinar on soils. It will be a cross-sector webinar on the 9th of December 2020, and we'll have presentations from researchers, applied researchers who've worked with farmers to reduce soil erosion. So really work must continue to save our soils and to keep them healthy. So this comes to, it's the end really of our webinar. Thank you for tuning in and have a good evening. Goodbye.